Let's see now the neuroglial cells or the glial cells. If you remember, these are the supporting cells. They also protect the neurons, but they do not conduct any action potential. They don't transmit any electrical signal. They only protect and support the neurons. And in general, we have two types of cells: Schwann cells and oligodendrocytes. Now, both cells perform the same function, which is forming the myelin sheath. So both cells form myelin sheath. However, the difference is that the Schwann cells are only in the BNS, in the peripheral nervous system, whereas the oligodendrocytes are in the CNS, in the central nervous system. This is the main difference between them. Now, what is the myelin sheath? The myelin sheath plays different roles. One of them is to save the neuron energy, but importantly, it speeds up the transmission of impulses. What does that mean? It means that when Schwann cells or the oligodendrocytes presented around neurons, these neurons usually transmit the signal faster. The action potential usually transmitted faster when there is myelin sheath. Okay, so they would speed up the transmission of impulses. Now, think of the myelin sheath as the plastic cover on electrical wire. You know, you have the electrical wire and there is usually the plastic cover over it. So this would preserve the electrical signal, also will protect it. So this is exactly how the myelin sheath is working. So again, the only difference between the two is their location. Now, these here on this image, these are the Schwann cells. This is a Schwann cell. This is a Schwann cell. This is a Schwann cell, Schwann cell, Schwann cell, okay? A Schwann cell. And as you can see here, these layers are the layers of the myelin sheath. These are the layers of the myelin sheath. And in the center is the axon. This is the axon. Now, because of the way, now notice that these cells, and again, this is in BNS, okay, in the peripheral nervous system. I want you to see how the Schwann cells are located. They are leaving gaps between them. There is some kind of a space. These spaces are called the nodes of Reinvard. These spaces, they are not covered with myelin sheath. Okay? So the signal, the electrical signal, technically jumps between these nodes, as you can see in this light purple arrows. So the signals is jumping from one node to another node, and instead of going through the entire axon. No, in this way, through this jump, the signal travels in a faster way. And that is the saltatory conductions of impulses in these neurons. Now, this is a video about the myelination and the saltatory conduction. Please watch it. It will help you. Now, this myelin sheath is so important that when it is degenerated, it will cause some diseases. A very famous disease is the multiple sclerosis. This is progressive damage to the myelin sheath in brain and spinal cord, and will result in weakness, visual impairment, and incontinence. All of these are symptoms of multiple sclerosis. Also, there is the amyotrophic lateral sclerosis, or the ALS. This is in a degenerative disease, or degenerative disease. Yes, it is degenerative disease, where the myelin sheath is degenerated, but in the motor area of the spinal cord. So it affects the motor area, the area responsible for giving the orders for our skeletal muscles. So here we will lose the skeletal muscles. Usually these patients will lose their ability to move, or to sit, or to use any of their limbs. With the MS, with the multiple sclerosis, it's more general for sensory and also for motor. But with the ALS, it's more of the motor area. So far we talked about the neurons, and we understood that they conduct the electrical signal. But we did not say to whom and how. To whom and how this electrical signal is conducted. Neurons have their own targets. These targets were, or the recipients of the electrical signals. So neurons, they have these targets and they deliver the signal to them. These could be another neuron, could be a muscle cell, could be a gland. They communicate with these targets through something called synapse. And synapse is a special junction between axon terminus and the target cell. If you remember the structure of the neuron, we said we have the cell body, the dendrite, then we have the axon and axon terminals, right? This is one neuron. There will be another neuron, could be another neuron. It has its own dendrites. I'll extend them a little bit. Then its own axon and axon terminal. Now, these axon terminals will form the synapse with the dendrites of the following neuron. So this is the synapse. So this is the junction between the axon terminals and the target cell. Could be a muscle, could be a gland. Now the transmission through the synapse is a complicated process that involves release and diffusion of neurotransmitters, chemical neurotransmitters, and that is what we are going to talk about. But before that, I want you to notice these two terms, the presynaptic neuron and the postsynaptic target, because this is a defining point. So this neuron is a presynaptic, and this neuron is a postsynaptic. So it is after the synapse. This is before the synapse, this is after the synapse. Now, the overall events happening at the synaptic transmission or during the synaptic transmission, we can list them that the action potential arrives at the axon terminus, causing calcium ions to diffuse into the axon bulb. Then the calcium ions will cause the release of neurotransmitter from the vesicles. Then the neurotransmitters will diffuse across the synaptic cleft. Then the neurotransmitters bind to receptors on the target and opens the gates. And once the gates are open, the sodium, usually these are sodium gates, would diffuse into the target cell. So let's go through these steps again. So the action potential arrives to the axon terminal. So the action potential already transmitted through the axon, and now it is at the axon terminal, so it arrives here. Once it's here, once it's in the axon terminal, it will open calcium gates. We have calcium outside this axon terminal. So once the gates are open, calcium will get inside. Calcium will cause these vesicles, these tiny vesicles, to come forward and release the neurotransmitter into this synaptic cleft. So this area is the synaptic cleft. I can't zoom in. So this area is the synaptic cleft. Now, within this area, these neurotransmitters will come and bind to the gates. Once they bind to the gates, they will open them, and sodium would diffuse or would run inside the neuron. would run inside neuron. Remember that the sodium outside the neuron, sorry, or outside the target cell, this could be as another neuron, could be a gland, could be a muscle. Okay, so the sodium outside the cell is higher in concentration comparing to inside. So once the gate is open, sodium would rush in. Sodium would rush in, and once sodium is in, it will cause the graded potential. If you remember the graded potential that we talked about earlier. So these are the 4 steps involving or summarizing the events happening at the neurojunction or at the synapse. Now, neurotransmitters are extremely important because they can either excite or inhibit the target cells. So the neurotransmitters, when released on the target cells, they can either activate or excite the cell, or deactivate it or inhibit these target cells. The action on the target cells depends on several factors. Depends on the type of the neurotransmitters. We have more than 50 types. Depends on the receptors, and depends on the type of gated ion channels on the target cells. Usually, and in general, excitatory neurotransmitters, the neurotransmitter that would activate the cells, usually will cause depolarization in the postsynaptic cells and causing the resting membrane potential to exceed the threshold. So usually excitatory neurotransmitters would lead to action potential, whereas inhibitory neurotransmitters will hyperpolarize the postsynaptic cells. Hyperpolarize, think of it, it will make it more sleeping. It will hyperpolarize, it takes the membrane potential away from the threshold. So here, no action potential can be induced easily when the inhibitory transmitters are released. This table shows several types of neurotransmitters. All of these are neurotransmitters, where they are released and their actions, whether it's inhibitory or excitatory. Sometimes some neurotransmitters could perform both functions depending on the receptors, like norepinephrine, acetylcholine, depending on the receptors. For example, acetylcholine is excitatory on skeletal muscles, but inhibitory at the cardiac muscle, for example, and so on. This is another video about the synapse. Please watch it. And maybe it's better to stop here and start a new video about the peripheral nervous system.